All right, welcome back, everybody. So, our next video is going to be covering chapter 4, which is protein. So, what I'm going to do is I'm going to have the PowerPoint up in the background. Again, I'm kind of sort of switch back and forth between that and the whiteboard. Again, some of the pictures, for some reason, my iPad's not scaling them properly, so they may go off scale. But if you have your PowerPoint on your real computer and or the photos in the book, while I'm doing this to look at, you should be in good shape. All right, so make sure to have those handy, and we're going to go ahead and get started. All right, so we just did chapter 3 talking about amino acids, and as we've mentioned, amino acids, they are considered the building block of proteins, right? They're the monomer that we use to make proteins by linking them in a big chain. So proteins, they are big chains of amino acids. It's not the only things that are necessarily present in proteins, but they're the main component. As we're going to see, there's going to be lots of important factors regarding both amino acids and then the protein itself that give a protein its identity, its properties, how it behaves, right? So the sequence is going to be important of amino acids. The three-dimensional shape, and there's actually lots of different aspects that go into determining the overall three-dimensional shape and arrangement within proteins. Sometimes you might have there are some proteins that can adopt multiple conformations, for example, right? And sometimes those can be altered, through some sort of external activity. A little bit of lingo. There are specifically protein conformations that have the ability to perform biological functions. The ones with biological activity are called native conformations. But we also can have some non-native conformations that can be brought about as well. All right. Several of the things that we're going to be looking at will involve patterns, various types of repeat patterns that continue to happen over and over again. We're going to be looking at a couple of different parts of proteins as well. We have the backbone, which is sort of the main linear component within the amino acid chain that sort of forms the core, the base of your protein. And then we have what we call side chains, and this is where the side chains of the amino acids are going to come in. So if you're talking about the backbone of a protein, really it's composed of the amino termini, carboxy termini, and the alpha carbons within amino acids. And then the side chains, the ones that are hanging off of the alpha carbons, I don't want to say they kind of do their own thing because that's not really accurate, but they can be thought of a little bit separately from the backbone. We'll see what we mean as we go through. All right, so you may have heard this before in biology classes. There's a few different types of quote-unquote structure that we can look at when analyzing protein activity and constitution, okay? And we sort of build from sort of generic, not generic, but sort of baseline type of things to some more advanced interactions that occur within a protein molecule. At the most basic level, the first thing we got to look at is what's our sequence of amino acids, right? A protein is a big old amino acid chain. The order of these amino acids is going to make a big difference as to what type of protein you have. Even in a small peptide, right? If you had glycine, valine, and histidine in that order from N-terminus to carboxy terminus N, it's going to behave differently than if you had them in a different order. I can't remember which ones I just named, so I can't give a sample, but you get the idea. Okay? So, the sequence of amino acids in your structure is going to be important. That's called primary structure. All right, the second level is going to be called secondary structure. And this is where we start getting more into three-dimensional arrangements. All right, this is going to be the 3D arrangement in space of the atoms that are in the backbone of your protein chain. So this is going to be, again, your N-terminus, your carboxy terminus, and your alpha carbon. All right, there's several different types of secondary structure. Couple of ones that you've probably heard of, and probably by far the most common ones, are the alpha helix and beta pleated sheet. All right, we're going to see that when we get these sort of basic secondary structures like this, there can be various ways in which these can repeat each other as well. So you can have, let's see, for example, a series of alpha helices that will repeat in a certain pattern, or beta sheets that repeat in a pattern, or a mixture thereof, right? And we have other terms for these as well that we'll get to as we go along. You sometimes have what we call domains. So you might have a protein that has secondary structure that is mostly alpha helix and or beta pleated sheet, but might have some other transitional or other, I don't want to say random, but maybe random, sometimes random, some sort of different pattern that's not really in one of those categories. How do we get our secondary structure? It's going to be from hydrogen bonding, and we're going to see that's a pattern, not only in proteins, but also biomolecules as a whole. Hydrogen bonding is a very important feature as far as keeping structural integrity and making sure things are aligned in a certain regular pattern. And that's something that definitely happens here. All right. Hydrogen bonding that occurs is from the amide H's, right? So you make the peptide bond within a protein. You have an NH group left over. There's one H there. That can hydrogen bond to a carbonyl oxygen. So, and we will look at those in more depth as we go along. These are just sort of introductions to the concepts. Then you have tertiary structure, and this is similar to secondary structure, but slightly different. It's a little bit more inclusive, right? When you're talking about the tertiary structure, you're not just talking about the backbone, but really you're talking about all of the atoms within the protein, right? All the atoms in each amino acid, including the side chains, which we don't necessarily include when looking at secondary structure. We also include other things that might be present. These are called prosthetic groups. These are atoms that are in the protein that are not part of an amino acid. A couple of really good examples of these are heme and hemoglobin, right? Hemoglobin's a protein that we'll look at a good bit later in the chapter. It's used to carry oxygen through the blood. And of course, it's got quite a bit of composition, of chains that have amino acid sequences, but you also have a heme group, which is an iron ion, an Fe, that is coordinated with something called a porphyrin ring. Very similar in chlorophyll, right? Chlorophyll present in plants. It's what makes them green. You have a prosthetic porphyrin group that has a metal in it that gives it sort of a green color, right? So prosthetic groups are essential oftentimes to the function and form of a protein. All right, and then you have quaternary structure, and this is not something that we look at in every single protein. Not every single protein has quaternary structure. Quaternary structure is when you have different large protein subunits that interact with each other, right? So if you have just one really long protein chain, right, one continuous amino acid chain into the forms of protein, and it folds up upon each other into a particular shape and all, you can describe its tertiary structure, but since there's only one chain, it doesn't have a quaternary structure. However, something like hemoglobin, it actually has 4 different chains, 4 different subunits that will arrange in a particular arrangement with respect to each other, and that's how we define its quaternary structure. All right? Cool. So we're going to take a look at these a little bit more, each of these a little bit more in depth. All right, so let's start off with primary structure. Primary structure is where it all begins, right? You got to have amino acids to build a chain, and it's just like putting a puzzle together, right? You got to put them in a certain order. The order that they're in will ultimately determine the secondary structure and the tertiary structure, and also if they're subunits, it'll contribute to the quaternary structure, because The sequence of amino acids determines where exactly atoms are located in space, right? So if you have an NH group in a certain spot in one chain, it might be able to hydrogen bond. But if the sequence is different, and if it's in a different spot in a different chain, it may not be able to form a hydrogen bond so easily, right? And that will affect the sort of physical and mechanical properties. Little things can make a big difference, all right? Really good example of this is a condition called sickle cell anemia. And I'm sure you've heard of sickle cell anemia. It's a pretty well-known condition, and it's probably also something that you've gone over in genetics because it is a genetic disease. Basically, what happens in sickle cell anemia is you have red blood cells, and they get kind of misshapen, right? So here you see a photo that has several normal red blood cells. You see they normally have that sort of disc shape, but then you see another one that's got kind of a crescent moon shape or a sickle-shaped cell as well. That's where they get the name from. And so sickle cells are not as efficient and not able to really carry and transport oxygen like normal red blood cells. And, of course, that presents a bunch of problems. All right? So, why the heck do we get sickle cells? Well, this is because of one substitution of one amino acid in one position in certain subunits of hemoglobin, what we call beta subunits, right? Normally at position 6 in hemoglobin, beta subunits, you have glutamic acid, all right? So, in fact, let me go ahead and pull this up. I can tell you, we can look at the first few amino acids in these hemoglobin beta subunits. All right. And so we're starting from left to right, as we always do. We're starting with the N-terminus N and going to carboxy. So the very first one in the chain is valine, then histidine, then leucine, threonine, proline, glutamic acid, and another glutamic acid. Oops. And sickle cell, it's very similar. Except for position 6. All right, so where you would normally have a glutamic acid, you instead have a valine. All right, and so if we compare the two, all right, I'm not going to draw the entire amino acid. I'm just going to draw the side chain because that's where they differ. All right, so these are the side chains of valine and glutamic acid, respectively. All right, so you can see they're very different. This one over here is basically a carboxylate group. It's got very electronegative oxygen and it's negatively charged, so it's quite polar. So polar it even has a full charge. Whereas this is an isopropyl group that is very non-polar. As we know, polar things are hydrophilic. Non-polar things are hydrophobic. All right. This makes a big difference because whenever hemoglobin subunits, whenever they fold and arrange themselves, they do so in order to align their amino acid side chains either towards one side or the other in alignment with water, right? So when you have that amino acid number in its number 6 position, it's going to be positioned much more in favor of alignment with other hydrophobic substances, whereas glutamic acid is going to be more aligned towards aqueous/water environment. Okay? What happens is, when you have these alignments happening within a sickle cell, let's see, what's the best way to represent this? I'm going to just do this with a very simple little cartoon, if I can think of what symbol I want to use here. Let's just use a squiggle. All right? So this little squiggle here represents valine side chain. Very hydrophobic, right? So what happens is you have these little valine side chains kind of poking out towards the surface of the cell where they normally would not. All right? And if you have several of them, several of these sort of, I guess, not properly formed side chain, or excuse me, hemoglobin subunits, what happens is these little things start sticking together, kind of like glue. A bunch of these can start gathering around and sort of start sticking to each other through what your book calls hydrophobic interactions. Guess what? That's just basically another term for van der Waals forces. Right? Those little weak interactions that we see that happen on all atoms, whenever you have a bunch of these present in these hydrophobic side chains and they sort of get clustered like this, they can start sticking to each other. And basically what happens is red blood cells, the hemoglobin polymerizes. So, hemoglobin itself is a polymer, right? Hemoglobin is a polymer chain. Basically what happens in this case, you get a polymer of polymers. You get a whole bunch of these polymeric hemoglobin chains kind of sticking together. Normally, that doesn't happen, right? So, when this happens, that's what causes the cell, whoops, I keep forgetting the swipe doesn't work on that. I have to hit this little button. So whenever that happens, it makes the cell a very odd shape. That's sort of that sickle cell shape, and it just doesn't perform its intended duties. Okay? So, that's certainly not the only example of this, but it's one really good and dramatic example, and I like to use it because it's one of the more well-known examples that a lot of people have mostly heard of. All right. Let's talk a little bit more about secondary structure. All right. So secondary structure, as we said, is going to be the backbones, carboxy terminus, N terminus, and alpha carbon. And secondary structure is highly dependent on hydrogen bonding. Specifically, and if you look at this picture, some of it got cut off, but you can see on one of the nitrogens you have an H clearly hanging off, right? This hydrogen is capable of hydrogen bonding, and it can form hydrogen bonds with oxygen coming out of the carbonyl carbon, right? So, as your primary structure develops and you have a sequence and your protein chain starts to form, this backbone can sort of be put into place when this H and those oxygens come into contact, and they can sort of, not bond with each other, but they can sort of stick to each other, right? And the pattern that they make as a result of this attraction that'll indicate what type of secondary structure that you have. Let's see. So, like I said, by far, can't talk today, the most common examples of these are the alpha helix and beta pleated sheets. And so these are examples of what we call periodic structures, aka secondary structures. And basically, they have a pattern, right? And that makes sense. Think of an alpha helix much like a telephone. Well, you guys are maybe too young to remember corded telephones. But if you look at my office phone, for example, I'm looking at it right now, it's got a wire on it. It's got a cord that's sort of coiled up, right? A very regular sort of helix structure. That's because in the alpha helix, you have regular repeating patterns that occur. All right, and we'll talk about those in just a second. This is possible because you have certain angular patterns that occur, bond angles that occur between certain atoms, right? And so these are called Ramachandran angles. And you can see these indicated in the photo by the psi and phi Greek symbols. All right? Let's take a little bit of a closer look at the alpha helix. So, how is an alpha helix made? All right, first of all, how many polymer chains are in an alpha helix? The answer is 1, right? So, each alpha helix is a chain unto itself. You can have multiple alpha helices bonded to each other or that interact with each other, but one alpha helix is one polypeptide chain. All right. These are stabilized by hydrogen bonds that are parallel to that helical axis. And there's a pattern here. What we tend to see is that carbonyl oxygens are hydrogen bonded to NH groups that are 4 residues away. Right? So, if you're looking at an amino acid sequence, right? And I'll just number them, right? 1, 2, 3, 4, 5. And I'm just drawing this as a curve, a very crude little cartoon or diagram, to sort of show you what happens. The alpha helix will twist in a manner so that you form those hydrogen bonds between 1 and 5, right? The first one is hydrogen bonded to a residue, an amino acid residue that's 4 units away. All right. 3.6 residues per turn. All right, and so this is an average. So every little turn where the coil forms, there's about 3.6 amino acids that sort of compose that turn. The exact number will depend on which amino acids are present, but it coils itself in a manner to where that's about right. And then the pitch, which you can see over here in this photo, basically corresponding portions of the same type of atom in different residues, there's a consistent length between those. It's 5.4 angstroms. So the alpha helix is very regular, right? It's very patterned. It's definitely got a rigid sort of set of rules that it goes by, right? Not to say, though, we can't have some variations. There are some things that could cause what we call disruptions to an alpha helical structure. All right? Well, let's take a look at a couple of these. One particular amino acid that can cause some disruption sometimes is the presence of proline. Why? Proline, look at the structure. So this is where if I had you in class, I would, start probing you for questions, but you're not here, so I guess I'll skip ahead. If you would like to ponder this before I tell you, feel free to pause this and look at the structure of proline. In fact, I encourage that. 3, 2, 1. Yeah, if you look at the structure of proline, it is a really unique amino acid structure. it's got this unique structure where the side chain actually sort of loops around and forms a ring by making a covalent bond to your amino terminus, right? It's the only one that does that. The presence of this five-membered ring creates sort of a bulky, unwieldy group that kind of causes like little kinks in the chain, so to speak, right? It can cause like little bubbles, little imperfections in the coil, in the pitch, right? So sometimes this doesn't really make much of a difference in practicality. Sometimes it can. It just sort of depends on the situation. But proline structure really lends itself to that. Electrostatic interactions. This is where you have, similar, like charges, they repel each other, right? Well, if in your coil you happen to have, let's say, a negative side chain that is coming into kind of close contact with another negative side chain, they're going to repel each other a little bit, right? So that's another thing that can sort of cause like a sort of a little kink or a little bubble, a little abnormality in the distance. We mentioned steric hindrance with proline, and there are some other ones as well that have not a five-membered ring to the amino terminus, but they do have some other big bulky groups on them. All right, so if you have a bunch of those that are sort of all in a row and very, very close to each other, they will start to kind of crowd each other out. This, by the way, disruptions does not mean problems or mutations or anything like that. It just is a disruption to the regular structure and shape. It means it doesn't look like a perfect helix. Doesn't mean it doesn't function perfectly fine, although there are some disruptions that occasionally can cause some problems. Again, very situational. All right, and you can see it got cut off a little bit, but you can actually see in this photo partially that you have several alpha helices that are sort of bonded to each other, right? And this happens quite a bit. We'll come back to that in a little bit. All right, the other really well-known one is the beta-pleated sheet. And so I've seen this oftentimes kind of represented in books, almost in photos. They look sort of like a corrugated tin roof or something that has a little zigzag pattern, or you can think of it like a skeletal organic structure, right? And then this book kind of represents it with arrows, so use whatever you want. But the basics of this state that, instead of having your amino acid chain kind of coil up like in an alpha helix, the chains do the opposite. They kind of extend, they kind of stretch out longer, and they form more of a sheet-like structure instead of a coiled helix. All right. Again, hydrogen bonds are going to be super duper important here. They're going to be what keeps the beta sheets aligned with each other in a certain arrangement. All right. We can have what we call intra-chain hydrogen bonds or interchain hydrogen bonds, and so interchain would be when you have two different beta sheets that are sort of interacting with each other. Intrachain happens when you have one long beta sheet that sort of folds over and doubles back and interacts and hydrogen bonds with another portion of itself. When you have beta sheets hydrogen bonding with each other, they can be in one of two alignments. It can be in what we call the parallel arrangement or the anti-parallel. This basically refers to how are the termini aligned, right? If you look at this photo right here, and again, part of it got cut off, so please refer to your PowerPoint on a real computer or your book, but we can at least see the A part here, right? So you see the little arrow, and you see the C and the N. This basically means here, like if I'm looking at this top chain structure, the amino terminus is on the right side, and it's as it goes right to left, you're going towards the carboxy terminus, right? If you look at the second structure, there's an arrow underneath it as well, and it's in the same direction. That means each of these two portions are parallel to each other. They're both going N terminus to carbon terminus, right to left. right? So this would be a parallel arrangement. The one on the bottom, which you can't see here, but you should be able to see in your other PowerPoint, is different. One of the chains is going left to right, nitrogen to carbon. The other one is going left to right, carbon to nitrogen. These are opposite, so this is an anti-parallel arrangement. You can make hydrogen bonds both ways, although the angle of the hydrogen bonds are straighter in the anti-parallel arrangement. How do you think that would affect the structure and properties of beta-pleated sheets? Maybe I'll let you figure that one out. Come back to me if you'd like to talk about it. All right. All right, so now proteins aren't boring. You know, they're not just going to be, well, most of them anyway. Most of them aren't just going to be individual alpha helices or beta sheets. They can make lots and lots of different combinations. I'm going to come back to reverse turns in a second. So, these are called supersecondary structures, aka domains. And there are buku-oodles of them. There's tons of them. I'll just show you a few. If you're a very aesthetic, design-oriented person, you might like this. Just to go over some of them real quick. Alpha-alpha. That's where you have two alpha helices that are connected to each other by some sort of connecting strip. And this usually something called a reverse turn, which I'll mention in just a minute. You can see it in part B of this photo. All right, that's an example of an alpha-alpha. You can have beta-alpha-beta, right? Intuitively, you would think that's a beta sheet, an alpha sheet, and another beta sheet. And you are correct. Specifically, a beta-alpha-beta is going to be two parallel beta sheets connected by an alpha helix. And you can see that in part A. And then you start getting into the really cool ones, the really fancy ones. You have something called a beta meander, and that's basically a series of anti-parallel beta sheets that are sort of connected by reverse turns going, doubling back on themselves, going in a consistent sort of snake-like looping pattern. You can't see it on this, what I'm showing you right now, but if you look in your book or on your copy of the PowerPoint, you'll see another one that's my favorite. It's called the Greek key. And the Greek key is another anti-parallel beta sheet arrangement, but instead of just connecting to the one next to you, they'll sort of randomly, or not random, they'll sort of skip sheets. So like number 1 might connect to number 3, number 4 might connect to number 2, etc., etc. And the reason it's called a Greek key is because the patterns that are made are kind of similar to the Greek key pattern. This is the name of the design that's on a lot of ancient Greek artifacts and stuff. So that's a fun little fact. But there's all sorts of different domains. Those are some of the more common ones. All right, let me talk a little bit more about the reverse turn now. So, oftentimes within a protein, you'll see a fairly sudden or sharp, turn in direction of your protein, right? We can see it with each one of these that are pictured in these domains over here, right? Your protein's kind of going in one direction, and then, it's going to loop and go the other direction, right? So, the amino acids that compose these, these little turns are called reverse turns. There's a couple of different ones. Type 1 and type 2 here, and I got my pictures cut off. I want to open my book so I can see it better. Give me 1 second here. Okay, there's actually 3 of them. So type 1, type 2, and type 3. All right, and you can see each one of these sort of makes a sharply defined reversal. And there's going to be certain amino acids that you tend to see commonly found in reverse turns. One of them is going to be glycine. Ask yourself, why the heck would glycine be likely to be found at a reverse turn? I encourage you to think about that for a minute. Maybe pause this and see if you can figure it out. Now I'm going to tell you. Well, what's the side chain on a glycine? It's an H, right? Glycine has the smallest side chain. It's literally just an H. So H's, of course, are very, very small. They don't take up much space. So if you have that tiny, tiny little side chain in here as you're trying to suddenly reverse direction, that makes it easier than trying to have something sort of big and bulky. All right? So glycine's really commonly found. The other one that's commonly found, and this one is in type 3, is proline. And you may wonder, okay, why the heck is proline there? Didn't we just say it was kind of a big bulky group that caused, irregularities in alpha helices? Why, yes, we did. However, you'll see here in that photo that the proline side chain is actually sort of off to the side here, so it doesn't really get in the way. But the angles within the rest of the proline unit that are made as a result of its side chain structure, they fit in well towards forming sort of a sudden turn. So the angular fit is better. All right. So glycines and prolines are often found in these sudden reversals. All right. So we've seen secondary structures of alpha helices, beta sheets. We've seen domains, which are these repeated patterns. And then we can go even further with it. We can have domains that then themselves repeat into larger patterns. All right, and these are called motifs. I'm not really that concerned about you knowing a whole lot of motifs. It's just kind of fun to include. There's a good photo on page 91 of the book that shows several of them. This one in this picture is called a beta barrel, and you can see it literally makes sort of a barrel-like arrangement. Otherwise, I'm not going to spend a whole lot of time on motifs. All right, so let's shift gears a little bit here. We're still been talking about secondary structure. As we go from secondary structure into tertiary, it's good to sort of talk about another classification we can make within proteins. Proteins generally can be classified as either fibrous proteins or globular proteins, largely depending on whether they're water soluble or not. And also the shape, of course, right? Lots of different shapes, but usually a protein will do, generally speaking, one of two things. It will either have itself sort of aligned around a central axis, or it'll be in a big old spherical structure or something kind of similar to a sphere, sort of like a wadded up sheet of paper, right? It's determined by a couple of things, and how it relates to secondary structure is the backbone folding, right? How the backbone folds up upon itself, that's going to go a long way towards determining whether something is fibrous or globular. And this also determines the physical properties. Is it water soluble? Is it tough? Is it squishy? Is it whatever, right? For example, so if we look at fibrous proteins, like I said, we have polypeptide chains that are organized parallel along a single axis. Well, what the heck does that mean? Well, here's one really good example, and again, you can't see it very well in this photo, but look at your photo in your book or something. Look at collagen. You guys have probably heard of collagen before, right? It's a structural protein. It's found in cartilage, ligaments, bones, teeth. People often use it as filler for filling their lips, make their lips look plumper or something like that. Well, if you look at the structure of collagen, and I'm looking it up in my book too since I can't see it right here. you'll see what I mean by having parallel arrangements along an axis. It sort of looks like a piece of corded up rope, doesn't it? Right? If you buy rope, you'll see that it has oftentimes little cords that are sort of twisted together to form a stronger, larger unit, right? And that's exactly what happens with the fibrous proteins as well. It's hard to pull and break a rope. You have to put a lot of force on it because of the alignment of the individual cords. Same thing here. Whenever you have fibrous proteins, like in this example, collagen is a triple helix. It's got 3 different helix chains that are sort of twisted amongst each other. The chains can interact with each other and provide lots of structural, a lot of intermolecular interactions that really make it hard to break up, right? This can happen with helices, as we see here, but it can also happen with sheets as well. You can have beta sheets that are aligned, sort of like the individual cards in a deck of cards, right? That gives it a lot of close interactions intermolecularly, and also gives it a lot of strength. Right, so we talked about collagen. Keratin is another common one. Hair is made out of keratin. Fingernails made out of keratin. Wool, a bull's horns are made out of keratin. All this stuff, as you can tell, is water insoluble. Wouldn't it be terrible if our bones and teeth were water soluble? That would be a problem, considering we are mostly composed of water. So that's what gives sort of fibrous proteins their properties. And then you have globular proteins. Like I said, these are more folded up into sort of a spherical shape. They're less regular and more random than fibrous proteins. Globular proteins, unlike fibrous proteins, are soluble in water and dilute salt solutions. And so the way they do this, they tend to arrange themselves kind of similar to a micelle, actually, in a way. The polar side chains that are present, they tend to be exposed to the outside, and that's where the water is. And the non-polar side chains tend to be sort of towards the middle, and these form little pockets. And as we go further along, we're going to see these little pockets are oftentimes where things can come in and bind. If you look at your photo of myoglobin, that's what this thing is. It's better in the book. You can see, again, kind of looks like a wadded up sheet of paper, in a way, right? Secondary structure is very important in helping to determine this, but also tertiary structure is important, all right? Tertiary structure is going to include a 3D arrangement of all the atoms, not only the backbone atoms, but also the side chains. All right, and so we're going to see, again, some common interactions that can occur between a variety, between atoms, and a number of non-covalent interactions. Hydrogen bonding is what? Hydrogen bonding of side chains, in addition to what we see with the hydrogen bonding of the backbone, right? So if you look at this photo, sorry, I've got the hiccups, we can see hydrogen bonds between a couple of different side chains to the right of center. Hydrophobic interactions. Again, if you have side chains that have non-polar groups, these hydrophobic interactions, aka van der Waals interactions, they can provide integrity to the structure of the protein. Electrostatic interactions, these are going to be charges, full charges. So if you have a negative carboxy group in one area interacting with a fully positive amino group, that would be an example of an electrostatic interaction. Complexes, this is stuff we've not talked about, certainly not in organic chemistry, but if you've taken analytical, you'll know that metal ions can bind and interact with a number of atoms themselves, and they form what we call complexes. So if you have metal ions that are present, those can also link a couple of chains. And one other one that is a covalent bond, not just an intermolecular interaction, is our old friend the disulfide bond. All right, and we mentioned this in chapter 3 when we talked about cysteine, right? We said cysteine had thiol groups, and these thiol groups could bond to each other to form disulfide groups. Or in this case, we call them disulfide bridges. And these are the only one of these that are actually covalent in nature, but they can help keep the protein chain in place. Okay. How do you determine tertiary structure? Honestly, I'm not really too worried about this with you because guess what? We don't have the equipment to do this. But there are a couple of different ways. There's one called X-ray crystallography that you can use. You can basically shoot a protein with a bunch of X-rays and it displays a pattern from which you can discern the three-dimensional arrangement if you're trained in it. And also, NMR spectroscopy, everybody's favorite from organic chemistry, right? Except it's a different type. It's called 2D NMR. Ooh, two-dimensional NMR. So it looks a lot different. And honestly, I just put this in there because I wanted to show you a 2D NMR. If you remember NMRs from organic chemistry, you're going to be totally confused by this ink blot thing over here. This is a 2D NMR. Again, it's kind of cut off a little bit. I used to read these. I don't know if I could still do it or not. It'd probably take me a little bit to sort of jog my memory. But again, like I said, I'm not too concerned about you knowing about this. I just kind of wanted to mention it. All right. Give me just a second here. I think I'm going to cut this off right here, and we're going to make this a part 1. Go ahead and be reading in this chapter. Obviously, hopefully you've been listening to this, so you're up to date so far. And I will get the second part up. Hopefully, my goal is to get it up tomorrow. All right, until then, have a good one. Bye-bye.